Okay, so in the last lecture, we learned that there are varying number of introns in a particular gene. Now, this is a very large RNA. Okay? Some portions have to be marked as introns and some portions have to be marked as exons. How is it done? And it is done every time the same way. Like imagine that if this is an intron, this is an intron. Sorry, something wrong. Okay, yeah. So imagine that this is an intron, this is an intron, this is an intron. And instead of excising out precisely like this, in some instance it is excised out like this, then all the reading frame and everything will be lost, right? So there has to be a precision exactly which part to be taken out and which part not. So we will first learn about that. So when you compare the sequence of the DNA with the cDNA, that is the DNA counterpart of the RNA, you can clearly tell that which parts are retained and which parts are not. So if you mark the exons, like this is an exon, this is an exon. In this figure, it is being called 5' exon, this is being called 3' exon. It only means that in the sense orientation of 5' to 3' in the mRNA, the exon that is more on the 5' side is being called 5' exon, and relatively that the one that is on the 3' side, it is called 3' exon. If there were another exon over here, then this will be called a 5' exon, and this will be called a 3' exon. That's irrelevant. Now, if you match it, so you will be able to tell where the exon ends and the next exon begins. And if you compare it with the genomic DNA, then you can see that just after the boundary of the 5' exon, there is always a G and an U. This is 100% conserved, okay, all the time. You will see that the first two nucleotides of an intron are a G and a U. The last two nucleotides of an intron is an A and a G. And somewhere between 20 to 50 bases, there will be an A, which is called the branch point. This is the only thing that is conserved, 5 nucleotides. Okay, a typical length of the intron will be, somewhere between 700 bases to the 3 kb, sometimes even longer than that. And within that, only this much will be conserved. And there is a pyrimidine-rich region, which is between the branch point and and this AG. So where splicing occurs, the junction between the exon and the intron, the 5' end of the intron, that is called the 5' splice site. The junction between the 3' end of the intron and the next exon is called the 3' splice site. So of this whole thing, only this much is conserved, and the remaining nucleotides, there is varying degree of conservation. Okay, so some breathing is allowed, but it is not that these 5 nucleotides are the only things that matter. The other things matter too, and you will see it through experimental designs. Okay, now how does splicing occur? So the way splicing happens is that this branch point A, the adenine, it initiates an esterification reaction with the phosphate group of this GU on the like the phosphate that is part of the diester, this phosphodiester, between the last nucleotide of the exon and the first nucleotide of the intron. That phosphate is attacked by the branch point. Okay, and then you see this is being attacked. So this is the first nucleotide of the intron. This is the last nucleotide of the exon is being attacked, and you get this kind of a circular structure. Kind of the 5' end is hanging out without any support, and then the circular structure continues into the 3' end of the mRNA. Then this hydroxyl group, which is created because of this esterification reaction, then attacks this phosphate group, which is bridging this nucleotide and this nucleotide, the first nucleotide of the 3' exon and the last nucleotide of the intron. That is, another esterification reaction happens, and then again the phosphodiester bond is formed, and now it is a continuity. Okay, and this little part excised out, and it is this circular structure is called a lariat. Okay, so the intron is excised out as a lariat. Okay, so it is fairly simple. Two trans-esterification reactions is what gives you the the splicing. Like you excise out the interim sequence. So, but I'm sure you realize that the process has to be very intricately regulated. You cannot just take out any sequence. Okay, it has to be always absolutely precise, and you will see some of the experiments, some data from the literature that will give you some idea. So now the splicing happens in conjunction with several small nuclear RNA or snRNAs. In splicing, 5 snRNAs take part: U1, U2, U4, U5, and U6. Okay, these 5 snRNAs, they do not act by themselves. They are in complex with a variety of proteins. Typically, there are 170 proteins that are, in complex with the splicing RNAs. There are other specific splicing factors which are not ribonucleoproteins. These are ribonucleoproteins. And the small nuclear RNAs, they are typically 107 to 210 nucleotide long. They are associated with proteins. So this is ribonucleoprotein complex. The spliceosome complex is almost as big as the ribosomes. So these ribonucleoprotein complex, they recognize different components of the mRNA for the splicing to happen. Okay? Now, U1 snRNA, it recognizes the 5' splice site, the exon-intron junction. Now, the experiment that was done, remember that this GU is 100% conserved. This A in the previous slide, if you look at it, this A is 70% conserved, not very conserved. But if you artificially mutate it, then splicing is blocked. And this splicing can now be restored if you artificially change this U to this G was mutated to an A, that's why splicing was blocked. And now if you mutate this C to an U, then splicing is restored again. So therefore, though only these 2 nucleotides are 100% conserved, but the nucleotides surrounding the splice site, they also play an important regulatory role. Like this A is not one of the conserved nucleotides, but it plays an important regulatory role. Otherwise, compensatory mutation would not have restored splicing, right? So, the way it works is that there is this U1 snRNA, then there is U2 snRNA that recognizes the branch point. See how the U2 snRNA works. Like it is complementary to a stretch in the mRNA around the branch point, but this A does not have a counterpart in the snRNA. As a result, the A remains bulging out, and that allows it to initiate the trans-esterification reaction into here. Okay, so branch point is recognized separately. The 3' splice junction is recognized by U2AF, and the snRNAs, etc. They all play a role in there, and then the splicing happens. I do not want you to get into all the complications of the process. I want you to understand it philosophically. If you are, working in a lab that is deep into splicing and splicing machinery, by all means, you should learn everything about splicing. By the way, I should inform you that there is a paper published in Science, I think 2004, 2003, 2004, something like that. The paper was about super conserved regions. Super conserved regions. These are stretches in the genome that did not change between bacteria to humans. Okay, if you do like sequence alignment in the DNA sequence of all sorts of organisms, transcending, like the evolutionary time, and you still find a stretch of DNA sequence longer than 200 nucleotide, that you call the super conserved region. So some of the splicing factor encoding genes, they belong to the family of super conserved regions, meaning that the proteins that are involved in splicing, they are very ancient proteins. So therefore, splicing plays a major role in animal physiology. There is no taking away of that. But all I'm saying is that for this particular course, I will not get into the intricate detail of splicing. That is not the purpose of the course. But if you are in a lab that studies splicing in that depth, then you should know everything about it. So, as I said earlier, and many times, that the CTD acts as a integrating platform. So the phosphorylated CTD, it depending on its splicing, like phosphorylation status, it can recruit many of the spliceosome components, and in turn, these heteronuclear RNPs, they enhance the interaction of the RNA polymerase II with the elongation factors like DSIF and CDK9 cyclin T, which is called a transcription elongation factor B, a phosphorylated transcription elongation factor B. It increases their interaction with the RNA pol 2, which further enhances the rate of elongation. So therefore, there is a link between CTD phosphorylation status, splicing, and the rate of transcription elongation. So we are coming back to the same theme over and over again, that how important it is to understand that CTD phosphorylation is a major feature of transcriptional regulation or even post-transcriptional regulation. Now, a little bit more detail into splicing, that how is it understood? Clearly, you can realize that those 5 nucleotides cannot be the everything about regulation of splicing. So there are intricate mechanisms by which the part of DNA is marked as exon. Okay, it is not that we fully understand exactly what it is, but certain sequence features in the DNA is known, and correspondingly in the primary transcript. Certain other proteins are known. So there are these sequences called ESEs, exonic splicing enhancers. Okay, and the proteins that bind to it, splicing regulator proteins or SR proteins. So because of the presence of the ESEs, the SR proteins bind to them, and the SR protein bound region of the mRNA, SR protein bound regions of the primary transcript are marked as exons. So they have to be retained in the mRNA. And there is an interaction between the SR proteins and some of the splicing factors like this U2AF and U1. So once SR protein is recruited in the exon, then they will recruit this U1 or U2AF in the nearby regions, And those are the 5' or 3' splice junctions, depending on where you are. Okay, so therefore, marking a stretch of primary transcript as the exon is critical in seeding which part will be marked as intron and will be spliced out. Okay, because these U1 and U2AFs, these are critical, effector molecules of splicing, meaning they carry out the process of splicing. Now, how do the sRNAs discriminate exons from introns? Of course, there are those ESCs, but that does not answer everything, and a whole lot is left for speculation. But one thing is very interesting, that whether it is the flies or the worms or the humans, majority of the exons are in the range of 100 to 300 nucleotide. While the introns, they can vary anywhere between 100 base pair to up to 30 kb. Okay, the human genes tend to have much longer introns than the fly genes or worm genes, but the bottom line is that the intron length is highly variable, while the exon lengths are quite consistent. And of course the ESCs. But you should know, first of all, that how the exons are discriminated from introns. The answer in full detail, I don't know. I do not think even the scientific community knows. But there are certain features. One is that the more or less uniform length of the exons, presence of the ESCs. Okay, there are two kinds of introns. One is group 1 intron, where they do not need a separate spliceosome complex. The group 1 introns, they themselves can assemble as spliceosome complex and help in splicing. And the group 2 introns, and the other is the ones that we learned about, where the primary transcript is there, and the spliceosome is assembled on top of it to effect splicing. But you can see that the overall structure, architecture of group 1 intron, please understand, group 2 intron, please understand that this is one contiguous RNA, okay, which is folded like this. This RNA has this secondary structure. So it is auto-splicing RNA. They do not need separately the splicing factors or they still need the splicing factors, but not the snRNAs, the U1, U2, U4, U5, U6 snRNAs. Because that is kind of built into the RNA primary transcript to begin with. Okay. The now splicing is done. Okay. Now, not done, it's just some basic ideas of splicing you got. Now, how is the polyadenylation done? So, as I said, that the actual length of the RNA that you see when you sequence the cDNA is the one up just before the poly A tail, right? So the poly A tail, you do not find it on the DNA because this is not template driven. This is after the RNA is processed, this A is added. But just before the poly A tail, that is, you will find it on the genome, genomic DNA. But the actual RNA is transcribed well beyond that point. So if this is your polyadenylation site, then there will be a signature AAUAAA, which is there upstream, typically 10 to 35 nucleotide upstream of the polyadenylation site. And there will be a GU-rich stretch, 50 nucleotide downstream of the polyadenylation site. And these two sites, they recruit two different kinds of proteins. The poly A signal, that AAUAAA, that recruits what is called CPSF, cleavage and polyadenylation specific factor. Independently, at this GU-rich stretch, CSTF or cleavage stimulatory factor is recruited. So this protein actually does the cleavage. So there is the endonuclease. And this protein, the CSTF, it stimulates the endonuclease activity. So you can imagine that why such a two-factor combo is important, because this stretch AAUAAA may be there in other locations on the mRNA. Okay, but unless that is followed, some 50, 60 nucleotides downstream by this GU stretch, then the polyadenylation will not happen. Our mRNA will not be clipped. Okay, so therefore this like a two-factor combo is needed. Once these two are recruited, then the cleavage factor 1 and cleavage factor 2 are recruited, CF1 and CF2. Then they finally recruit polyapolymerase, PAP, polyapolymerase. Now, once this assembly is there, then the RNA is cleaved at the polyadenylation site. So you can see that the hydroxyl group is exposed now. To this, polyapolymerase will slowly add 12 nucleotide around 12 A's, which is the initial part of the polyadenylation. After that, polyapolymerase can add up to 250 nucleotide, 250 A's. Okay, but this part is promoted by another factor called PABP2, polyadenylation, poly binding protein 2. So PABP2 actually stimulates the polyadenylation by PAP. Now, this is a regulatory step. Okay, it is still not known that what dictates that it will stop around 250. In certain mRNAs, it stops around 50. They are less stable. Okay, so exactly what dictates, regulates the length of the poly A tail, we fully do not understand. But we do know that it's a major regulatory step when it comes to RNA stability in vitro. Okay, I would strongly encourage you to read this review article. It is a relatively old review article, but it gives you a very, very good, comprehensive concept of transcriptional regulation up to post-transcriptional regulation. It is a strongly recommended review article. Okay. Now, I will end this part with a very classic example of alternative splicing. Okay, alternative splicing meaning that you may have an mRNA. You may have an mRNA where there are, exons, many exons and introns. Okay, so this is exon 1, exon 2, exon 3, exon 4, exon 5. Now, you may think that all these exons will be present in the final mRNA, which is almost never the case. These exons are like the superset. If you remember your class 12 level probability, like combination, computation combination questions. So you have this repertoire of 5 exons. You may have in the final mRNA any of the 4. Okay, so if it is any of the 4, then the rule is that the first exon is almost always there. Okay, so the remaining 4, any 3. So the second one can be in any one of the 4 ways. The third one can be in any one of the 3 ways, and the fourth one can be any one of the 2 ways. So 24 different variations you can get if you only want to have a 4 exon mRNA coming from this transcript, which has 5 different exons. And I gave you the example of DSCAN, which can be reorganized into the 38,016 different mRNAs. Okay? So now this differential splicing has many regulatory implications. I will give you some examples. But the most classic one that is well studied is drosophila sex determination. Drosophila sex determination is not exactly like ours, okay? But the basis is the same, is the number of X chromosome is what dictates it. What is the end point is this protein, DSX protein. This DSX protein is of two types, the male type DSX protein or a female type DSX protein. So whether you will be making the male type DSX protein or the female type DSX protein will dictate the sex of the embryo. Okay, and that is dictated by the X chromosome is to autosome ratio. Okay, in females there are 2 X's, in males there is 1 X. Okay, and autosome of course is 2A. So this X chromosome is to autosome ratio, how that because for us it is the Y chromosome that imposes the male fate on an otherwise default female sex. Okay, so do not take my words too seriously. I know very little about sex determination, particularly in humans, but in humans, it is, the only job of the Y chromosome is to impose male fate. But in Drosophila, that's not what it is. It is the ratio of the X chromosomes to autosomes that dictate that what will be the sex of the fly. And that happens through a series of differential splicing regulation. So the way it happens is that there are certain proteins that are produced from the X chromosome called cis A, B, etc. These are referred to as the numerator proteins. Numerator proteins, like in an equation, like what is there in the numerator, this is denominator. And from the autosome, certain proteins are produced, like deadpan is an example, which is a denominator protein. Deadpan is a typical inhibitor of this cis A, B, etc. proteins. What proteins those are? Don't bother. Like those are some kind of transcriptional regulators. Now, in females, because of 2 X chromosomes, the dosage of the numerator protein is similar to the dosage of the denominator protein. But due to affinity issues, there are some numerator proteins that are homodimers, and some are, of course, engaged in heterodimer with the denominator protein. But in males, they are always engaged in heterodimers. So therefore, they cannot bind to the early promoter of a gene called sex lethal. Okay, remember sex lethal has a RNA binding motif. Long time back, I told you in the last lecture, I told you. While in females, these numerator proteins can bind to the early promoter of sex lethal gene. If it binds to the early promoter of the sex lethal genes, some sex lethal proteins are produced. Okay, this part is spliced out, and this is produced. Now, irrespective of the sex or this ratio, the late promoter is recognized both in males and in females. If it is transcribed from the late promoter, then this exon, the start codon is within it. This exon and these exons are produced. Okay, please note that this exon has a in-frame stop codon. In-frame stop codon. In-frame with this, like after you whatever the leading frame is, there is an in-frame stop codon. Now, if this sex lethal protein is there, then it acts as a splicing factor to recognize this as an intron, like to regulate, so that this is spliced out and you actually make a sex lethal protein in the females. While in males, you have this exon also as part of the mRNA, and therefore there is a premature termination of the translation, and you do not have a functional sex lethal protein. Okay, so sex lethal protein basically, therefore, is a splicing factor. It acts to remove the male exon from the transcript in the females. Therefore, sex lethal protein is produced. Once the sex lethal protein is produced, so in female, sex lethal protein is produced. In male, it is not produced because of this premature like translation stop codon. Then this sex lethal, for another gene called TRA, this sex lethal again does the same thing. It blocks splicing of exon 1 to exon 2. And instead, splicing happens directly with exon 1 and exon 3. Okay. So, but again, in males, exon 2 of TRA protein will be included. That has a premature stop codon, so TRA protein will also not be made. Now, this TRA protein is like an SR protein. Okay, it binds to the exonic splicing enhancer and then recruits RBP1 and TRA2, two more proteins to itself, and then exon 4 is recognized as an exon, therefore only in females, and that has its own polyadenylation sequence. So in the female, the drosophila SX, the DSX, ends at exon 4, and you have the female specific DSX protein. In males, because this TRA protein is not there, so exon 4 is not recognized. Therefore, exon 3 spliced directly to exon 5. So this is, like the exonic definition did not happen. So exon 4 is not defined as an exon in the males. So therefore, you will make a different protein. Like I think the mRNA actually goes beyond exon 5 because the polyadenylation sequence is not there, but it's a different protein. So this is how sex determination works in Drosophila by alternative splicing. As I was saying that, this TRA protein, this RBP1 and TRA2, they can bind to exon 4 only if TRA protein is there. Okay. Similarly, now we know that differential splicing is critically important for our hearing. Okay, it's interesting that in the inner ear there are hair cells, and these hair cells, they have the they respond to different frequencies of sound. Okay, there is a polarity, there is a apicobasal polarity in the hair cells of the inner ear, and they respond to different frequencies of sound. And the way we hear is basically purely electrical, it's a matter of electricity flow. So once the hair cells are excited, then they activate some potassium channels, which is calcium activated potassium channels, and then neuronal depolarization activated with specific kinases, and that's how we get to hear. Now there are some SR proteins and HNRNPs that are differentially post-translationally regulated because of these protein kinases, and they, alternatively splice, proteins involved in the ability of hearing in the hair cells, and so here the clarity of hearing that we have is basically dependent on alternative splicing. Okay, if you want to know the detail of it, I am really not the person. You should ask Dr. Janaki-san. She is an expert. She will be able to tell you the exact detail of how these proteins act and etc. But my job was to alert you to the fact that your clarity of hearing is dependent on alternative splicing. And of course, we know about DSCAM. And whatever we have said so far, none of that can explain how so many different kinds of splicing, like exons are recognized in so many different combinations, and every time the right combination in every cell type. But just to make you understand it clearly, that in drosophila it looks like almost all the alternatively spliced forms are important in the wild type flies. Therefore, DSCAM can undergo alternative splicing in 38,016 different ways, and they are needed for the normal cellular and organismal physiology. What it means is that these 38,016 different types of splice variants are precisely produced depending on the cell type and or other physiological parameters. And of course, we do not understand how exactly it happens. Apart from splicing, there is another way of RNA modification, which is extremely rare. It is called RNA editing. Okay, this particular gene, apolipoprotein B, in liver, there is this codon CAA. In intestine, this CAA, it is a post-transcriptional regulation. Okay, the cytosine is converted to a neurosil after the mRNA is produced. Okay, and there the CAA is changed to UAA, so you have a premature termination codon, and you produce a 48 kD protein. While in liver, you produce a 100 kD protein. It is an extremely rare thing, but it does happen. Okay, so with that, I will end it here today, and in the next lecture, we will focus on RNA transport. Okay, thank you.